Good morning, everybody. First, let me introduce myself. I'm Charles RODDE, and I am R&D manager at Entofood. Entofood is a company that is producing insect, meal, and oil, in particular, from black soldier fly. My presentation of today will be about antimicrobial activity and immune enhancement effects of insect, meal, and oil. So, the questions I'm going to cover today are these ones. Firstly, I will explain you why finding an alternative to antibiotics to tackle bacterial infections is clearly a matter of life and death. Then, secondly, I will explain you why insect products, meal and oil, could be an alternative to antibiotics. Finally, I will detail to what extent insect products enhance immune system of farmed animals. So first, to give you the background, there is currently an overuse of antibiotics. Firstly, in human beings. Between 2000 and 2015, the consumption of antibiotics has raised by 65%, and between 2015 until 2030, it is expected to raise by 202%. We observe the same trends in farmed animals. In terrestrial farmed animals, between 2010 and 2030, the consumption of antibiotics is projected to increase by 67%, and in aquaculture, the consumption is expected to rise by 33% between 2017 and 2030. In both cases, a large share of antibiotics consumption is questionable or not really necessary. And this overuse of antibiotics is a major threat to public and environment health. Why? Because when there is an overuse of antibiotic, there is a selection of bacteria that are resistant to antibiotics. For instance, when we use an antibiotic, all the bacteria are going to die except a few ones that are resistant to antibiotics. And then these few ones, they are going to multiply and the infection is going to strike back. There is the emergence of an antimicrobial resistance. So these bacteria can't be killed anymore with classic antibiotics therapies. Currently, these resistant bacteria are killing 700,000 patients per year, and by 2050, they are expected to kill 10 million patients per year. One of the most dangerous among the resistant bacteria is Streptococcus pneumoniae, that is causing deadly pneumonia. This overuse of antibiotics is also affecting the environment, in particular soil microorganisms, plants by decreasing their photosynthesis capacity, and water organisms. So finding an alternative to antibiotics to tackle bacterial infections is truly a matter of life and death. And now you may wonder, what is the link with insect products, with insect meal and insect oil? Firstly, to present a bit the context of insect farming, insects are currently an emerging source of food. For instance, if we have a look at the insect protein market for animal feed, in 2020, the volume of production was 10,000 tons. And this volume of production is expected to be multiplied by 50 to reach 500,000 tons per year in 2030. Main species reared are black soldier fly, Hermetia lucens, and mealworm, Tenebrio molitor. In both cases, the larvae are used in the process, produce insect meal and oil, and the adults are used for reproduction to get new larvae. And insects are seen as a sustainable, much more sustainable alternative to meat. Why? Because to produce 1 kilogram of insect, greenhouse gases emission are going to be much lower than in pigs and beef. Ammoniac emission is going to be far, far lower than in pigs. And the surface use, the land use, is going to be also much lower than in pigs and beef. Insects are also very nutritious for farmed animals. If we take the example of black soldier fly larvae, we have 40 to 50% of proteins, including essential amino acids, such as lysine, leucine, phenylalanine, 25 to 35% of lipids. Most present one are lauric acid, palmitic acid, oleic acid, 8 to 10% of chitin, and finally 10 to 20% of minerals, including manganese, calcium, iron, for instance. So this is the nutrition aspect. Insects are nutritious, but not only. Several components within insects, in fact, have an antimicrobial activity. The first one is within proteins, antimicrobial peptides. I'm going to detail the definition to be sure we are all on the same page. Peptide is a short chain of amino acid. It's in fact smaller than a protein. And antimicrobial peptides are effector molecules produced by the immune system, for instance, the immune system of insects, to combat microbes. Insects exhibit the greatest diversity of antimicrobial peptides within the animal reign. In total, more than 150 insect peptides with antimicrobial effects have been identified. Here are a few examples based on black soldier fly. The first one is insect defensin. It is the largest group of antimicrobial peptides, very, very widely spread among insects. We find it in Diptera, such as flies, in Coleoptera, such as beetles, in Lepidoptera, such as butterflies, in Hymenoptera, such as ants and bees, and these are only a few examples. Other antimicrobial peptides are cycropine, atacine, sarcotoxin. Antimicrobial peptides action, in fact, is to bind to bacterial cell and progressively destroy it and kill the bacteria. They are effective against a wide spectrum of bacteria, including Escherichia coli, Staphylococcus aureus, Salmonella species, Aerococcus viridans, that is accountable for heart and urinary diseases, Klebsiella pneumonia, that is leading to pneumonia, and these are only a few examples. Another component that has an antimicrobial activity is the lauric acid within lipids. Lauric acid is a saturated fatty acid, and it has also a derivative called glycerol monolaurate, that is in fact lauric acid plus a molecule of glycerol. Outside of insects, these molecules are mainly found in palm oil and coconut oil. In black soldier flies, lauric acid can represent up to 60% of fatty acids. The action of lauric acid and derivative is to destabilize bacterial cell membranes, so it is becoming permeable, to inhibit bacteria membrane exchange and to inhibit the energy metabolism of bacteria. To make it simpler, all of this is leading to bacteria death. Moreover, and this is a very important point, bacteria are unlikely to acquire resistance against lauric acid and its derivative. So lauric acid and its derivative have demonstrated effect against a wide range of bacteria, such as Escherichia coli, Staphylococcus aureus, Heliobacter pylori, that is accountable for gut diseases, or Cutibacterium acnes, that is responsible for acne on skin. The last antimicrobial component in insect is chitin. Chitin is a polymer that is a component of the exoskeleton of insects and crustaceans. For instance, on the larvae on the right, what you see is the exoskeleton, and it is mainly made of chitin. Chitin is a polymer, so it is forming a long chain. The molecule you see is repeated a very high number of times. Chitosan is produced commercially. It's a derivative from chitin, a commercial derivative, and both have the ability to react with bacterial cell membrane and damage it. Briefly, the positive charges of the nitrogen group of the molecule can react with the negative charges of the cell membrane of Bacteriae, making it more permeable and ultimately leading to Bacteriae death. The chitin and chitosan have demonstrated antimicrobial action against Escherichia coli, Salmonella species, Vibrio cholerae, accountable for cholera, Shigella dysenteria, accountable for dysentery, for instance. So insect products have demonstrated antimicrobial effects. But moreover, beyond these antimicrobial effects, insect products also promote various immune mechanisms in farm animals. The first and most investigated one is the effect of insect products on gut health. Insect products are promoting gut health in various ways. The first way is that insect products, and when I say insect products, I mean insect antimicrobial peptides, as well as insect oil and chitin, are able to shape the microbiome of farmed animals. First, their antimicrobial action is going to decrease the abundance of pathogen bacteria, and they are also able to increase the abundance of beneficial bacteria that can be called probiotics. For instance, the abundance of lactobacillus and bifidobacterium are increased. It is also the case, for instance, for ruminococcus, that is very important in ruminant animals. This microbiome shaping effect has been demonstrated in various species, such as pig, rabbit, turkey, Siberian sturgeon, rainbow trout. Another aspect is that insect products have the ability to reduce the inflammation within the gut of farmed animals, such as pig and turkey, making the digestion process easier. Finally, insect products permit to improve the mucus protection within the gut. In fact, the gut is secreting mucus to protect itself to form a barrier against pathogens. And when animals are fed with insect products, the mucus protection is improved. First, in quantity, more mucus is produced, and then in quality, the mucus is more concentrated in antipathogenic enzymes. It was demonstrated, for instance, in chicken, pig, and rainbow trout. Other immune mechanisms are promoted by insect products. The first example is in fish. Again about mucus, but this time not gut mucus, but skin mucus. Fish are producing mucus on their skin to form a barrier against pathogens. And it was observed that in Nile tilapia fed with insect products, the mucus has an improved lysozyme and peroxidase activities. Lysozyme and peroxidase being two enzymes with an antipathogenic effect. Another example is in chicken. Chicken, when they are fed with insect products and challenged with a virus called avian infectious bronchitis virus, are exhibiting an improved proliferation of T killer lymphocytes. So insect products are promoting the proliferation of T killer lymphocytes that are white cells going to directly attack and kill the viruses. And this effect is not neglectable because from an overall point of view, the mortalities of chicken are decreasing by 20%. We have a bit the same case with common carp, fed with insect products and challenged with Aeromonas microphylla, that is a bacteria. Common carps fed with insect products are exhibiting an improved proliferation of leukocytes that are white cells also. To conclude this presentation, what you should remember is that overuse of antibiotics and emergence of antimicrobial resistance are a threat to public and environmental health. Insect products comprise various antimicrobial components such as antimicrobial peptides, lauric acid, and chitin that could be an alternative to antibiotics. And insect products can also promote immune mechanisms in various tissues, such as gut, skin, and blood, but further investigations are still required to completely understand the full action of insect products. Here are the references I used if you want to investigate in deep this subject. And I would like to thank you for your attention.
Hi everyone, my name is Rafael Hermes. First of all, thank you for the invitation on participate on the vConnect. Also, thank you, Charles, for this first presentation that introduced pretty well the topic of antimicrobial and the effect that the insect products can have for different species. Today, we're going to talk about how the insect products can boost also the growth, performance, and immune system of aquatic species, mainly for a trial where we did with white leg shrimp. We have prepared this presentation today where we're going to cover, first of all, the part on how we can show the effect of antimicrobial of different insect components such as AMPs, lysozymes, and lauric acids. Our presentation will be divided into two parts. The first one is this in vitro antimicrobial activity, where we're going to talk about the inhibition growth at Petri dishes, where we test our products in 3 different pathogenic bacteria, and also an in vivo shrimp trial feed, where we tested the life performance, the survival rate, the immunity, and also the economic analysis. So let's start on the first part, where we did a very simple test in our lab, where we grow 3 different types of bacteria: E. coli, Salmonella, and Staphylococcus aureus, potentially pathogenic for all species. And we compare with a positive control bleach, as you may know, a very strong sanitizer and sterile water. And you can easily see that there was a big difference on the inhibition zone diameter when you use bleach, which means we confirm that it has a strong growth inhibition on these 3 bacteria. And then on the upper part, you can see that overall the BSF larvae and the fresh larvae can have an effect on the different effects on this bacteria. And also we tested very interestingly the insect oil versus a virgin coconut oil. The insect products stay halfway of the positive control and bleach, having a inhibition zone diameter at around from 0.6 to 1 centimeters, and the bleach was 1.6 to 2 centimeters. Now let's move on to the other project. There was a in vivo project that we have conducted in Vietnam at the Shrimp Vet Research Center from July to September. So the trial finalized this month. Is a very fresh data from this excited project that we developed with them in Vietnam, where we tested the BSF meal on the performance of white leg. The objectives of this project was, first of all, test what is the best cost benefit. So it's a dose response trial where we replaced at different levels the fish meal in the diets of white leg shrimp, right? And then we tested, also we assessed the life performance of this BSF meal in commercial feed. It's important to mention that this project was done with the help of a local shrimp industry in Vietnam. So it was done with ingredients that you can locally find in Vietnam with the levels that industry uses there, and also the fish meal that is currently being used in Vietnam. We also, in this trial, we evaluate the effect on immunity and how we can make a link between the antimicrobial and the immunity system. We're going to show you later. and also finalized by studying the economic feasibility of this use of this project by our shrimp industry. In the trial design was a very simple 5 different treatments where we included different levels of our product called high protein, which is a black soldier fly meal that contains 50% of crude protein. So we were replacing, you're going to see in the feed formulation dynamics how we replace the fish meal and what we have to include and take out from the formula. We tested with the 7 replicates. The shrimp size at the beginning of trial was around 1.18 grams. And we use in each tank, 350 liters tank, 40 animals. The trial lasted 44 days, including the 2 days of acclimatization. So the whole trial was a 6-week trial. Here you can see the nutritional levels that we use. So in all different diets, we use the same, we take very good care to formulate all diets with a 39% crude protein, which is pretty high. Lipids, everybody the same levels, lysine, methionine, threonine. So all diets were isoenergetic, isonitrogenous. We also took care to add cholesterol at that minimal 1 grams per kilo for all the different treatments, phospholipids, calcium, and phosphorus. So basically, in theory, all the different treatments, they have the same nutritional levels. That is very important to mention and to bear in mind. When you look into the feed formulas, you can see how we did that, right? So first of all, we use a premium fish meal, anchovy fish meal, a very premium with a 67% crude protein at a very high cost. And then we combine soybean meal and high protein to replace the fish meal. So you can see how you include the different levels of high protein, the insect meal, and how we combine with increasing levels to fill the gap of protein. Obviously, the fish meal had 67% of protein in our product, 50%. So we needed to cover with also a bigger inclusion of soybean meal. The insect meal usually contains a higher content also of fat. So what happened in the feed formulation was that the tuna oil was taken out from the formulas. Wheat flour was pretty much the same for everybody. We, of course, BSF meal, it does not contain cholesterol as the tuna oil and the fish meal. So we had to supplement cholesterol to the different diets, different treatment diets. Soy lecithin was included to correct the phospholipids. With that, also we had to include more amino acids to cover the gap that fish meal replacement caused in the diets. So this formulation was done in conjunction with the local industry, so was reviewed by local industry in Vietnam. So it's very much reflect what we can find in Vietnam currently. Here you have some pictures on the experimental diets. You can notice that with including higher doses of high protein, you have a whiter feed. There is absolutely no problem with that. Just like the feed is going to be lighter. It's just a visual observation. When we go to the water quality parameters during among treatments and the whole trials, we didn't observe any difference between the different treatments, the different parameters that we measure. They are absolutely under very normal conditions. So we can conclude here that the insect meal, replacing insect meal, the fish meal by insect meal, we cannot have like a negative or any difference on the water quality of shrimp. We brought here some pictures to illustrate. So on the left corner here, you can find the shrimp at the end of the trial from the control, where we can see the shrimp was around 11 centimeters length. And then we compare the control with 5%. So you can see how the different treatments, the shrimp is getting bigger. The biggest one was selected on the last treatment that includes more high protein. So having reached here around 13 centimeters, sorry, and with the highest inclusion level. When we see the live growth performance of this trial, at the beginning, of course, we didn't have any difference, but if you see the individual weight or the individual data on the animals that we selected, we sample from to take the measurements, there is no difference between treatments for final weight gain, for individual average daily gain, and individual specific growth rate. So everybody was the same. However, when we look into the survival rate, was the most amazing result we got with the high inclusion levels of high protein. We got the highest survival rate of around 97% for the 25% replacement compared to controlled 84. So it was a linear and positive response by including higher levels of high protein in the diets for shrimp. When we see the feed intake, here is the feed intake. Important to mention grams of feed per tank. We didn't observe any difference, significant difference between treatments, so there is no difference between them. Of course, if you divide it by the number of animals, in theory, these animals they ate less individually because we had more animals per tank, but overall everybody is around 500 grams of intake during the whole period, during the 42 days period. And then when we see the live yield, so grams of shrimp per tank, it's also an amazing significant difference between treatments. The control was 429 grams per tank compared to the 25% replacements as the highest one with more than 500 grams of live yield per tank. Also, again, a linear and positive response by replacing the fish meal by insect meal. For the feed conversion ratio, of course, as we observe, a higher live yield was also a linear response where we got better feed conversion ratios at 25% replacement, going from 1.43 to 1.19. So amazing results for the industry. That is what a local industry also is looking for. Is their main goal is to reach feed conversion ratios lower than 1.2. So this is an amazing result that can illustrate the effect of the product in the field. When we took some samples to measure some immunological parameters. In this case, we tested phenyloxidase is an enzyme that is responsible for the immune response. There is absolutely no significant difference between different treatments. We didn't observe any effect on phenyloxidase. Also, for the total hemocyte count, every treatment was the same. But what we noticed with these two data is, take a look, please, on the variability, right, on the CV. So it's higher in the control group compared to the other treatment groups, which means we have a more homogeneous, let's say, response for the animals for the phenyloxidase and total hemocyte count. When you look into the differential hemocyte count, where we measure the percentage of semi-granular, granular, and hyaline cells, there is no significant difference for semi-granular. However, we notice a tendency for both of these parameters to have, when we include higher doses, higher replacement levels, we notice that the granular cells were reducing and the hyaline cells was increasing. So the granular cell is a response for the immune system, and the hyaline is mainly for the phagocytosis. So it looks like our product stimulates the phagocytosis and natural immune response, so boost the immune system. And this is also can be related with this antimicrobial function and the chitin that can boost the immune system. That's what we believe that is a plus that this product can do for the animals. So it's not a common ingredient that just replaces protein, but also can have an impact on the immune system. We also have done in this trial we repeat over exactly the same trial with the same treatments over again, but now we use a different facility where we were able to challenge the animals with a bacteria called Vibrio parahemolyticus. Is a bacteria that is very pathogenic for shrimp and cause acute hepatopancreatic necrosis disease. So that's why we use this control environment where we have a different treatments. We have a special aeration system, submerged biofilter to control the disease and control the challenge. And the results we got, so on the survival rate, what we noticed when we compare the negative control here means there is no challenge with the challenge one, with all the challenge groups. So we saw a big impact on the survival rate. So it means apparently no treatment were able to kill the Vibrio. So to control the Vibrio is very pathogenic. So it worked the challenge, we can say. But when we look and compare the different treatments with different fish meal replacement levels, we notice that overall there is a tendency to have a stronger shrimp with this treatment, right? So 1/3 of this group survived to this very strong challenge. So it's again another indication that the product the insect meal can bring stronger animals and can boost the immune system to deal with very, very pathogenic bacteria such as Vibrio for shrimp. So that was the survival rate data. And we also did the economic analysis. So when we compare the feed cost, of course, these products today, the insect meal, the industry is growing, but it's not price competitive as fish meal. So the higher inclusion levels of fish meal we also incremented the price. When we compared the 25% replacement, which was the best live performance result, we are talking about the investment of $26 per metric ton. And when we start looking into the feed intake, was everybody the same, around 500 grams, but the live yield was different. If you remember, I present to you, especially this treatment was more than half a kilo per tank. So when we divided this number by the feed intake, you can have the feed efficiency, right? So feed efficiency means kilos of shrimp, you can get it from 1 kilo of feed. So obviously this treatment 25 was the highest feed efficiency, so was the most efficient one. And when we look into the revenue that this can generate, assuming here a price that this shrimp can be sold at $5 per kilo, and assuming 1 metric ton of feed, what is the revenue comes from this yield? We have a different revenues, but here especially this treatment was more than $600 per ton, so a nice yield. And when we divided this number by the investment, we can have what we call the return over the investment. So we can conclude here that especially this treatment, we got $24 back for every dollar that was invested in this nutrition. So it's amazing return over the investment that the industry can benefit. And yeah, so concluding our presentation, coming to the end, in this trial, we can conclude that first of all, BSF meal is able to improve the resistance of animals, and with that, we can increase the life performance We have the best cost benefit was including 6% of BSF meal, so means 25% fish meal replacement. Investing in this case only $26 per metric ton on white-legged shrimp. The farmers can take it back $609, so a return over the investment of 1 to 24, which is amazing. And the last conclusion we can have from this trial is that insect products are here. Our industry is growing. We can produce locally. We are producing in Southeast Asia. We can be very close to the industry and using the food waste, using sustainable ingredients. So by itself, our industry is sustainable. The animals we have a nice response on the live performance. We can prove that it's feasible. And the most important thing is that we noticed during this MCO, during this COVID pandemic situation, the price was fluctuating of the raw materials fluctuating during the year, but with our product, as we do not depending on the commodities, we can have a fixed price during the whole year, so no seasonality. And with that, I would like to thank you again. Here is my contact numbers. We are glad to listen to your question. Please visit us if you want to know more about this excited industry. And yeah, Nick and I, we are now open to listen to your questions and listen and answer as much as we can. So once again, thank you very much. Hello.
Hello, Rafael.
Hello.
So for the first question, I can answer if you want.
Yeah.
But I don't know, we are alone or?
I think we are live on the room, so you can answer the first question about the AMR.
Yes. So the impact until now, we have from literature there is no evidence that we can really using nutrition target the production of specific antimicrobial peptides. So we still don't know if it's possible. It doesn't mean that's not possible. We still have no evidence about it. Further work will be needed to solve this issue. Then from the very last results, what we know, however, is that probiotics can change the fatty acid profile, for instance, and they can increase the abundance of, for instance, lauric acid that has antimicrobial effect. So yes, probiotics, they could tend to improve the antimicrobial effect of insects, but further investigation is still required.
Good. Do we have more questions for us in the chat? If the organizer can send us the questions. No more questions for now. The question is if you have any information in poultry, chicken meal, I suppose, is broiler chicken, right? Do you have some information, Charles, about the antimicrobial effect in poultry?
Okay, first, the first point is, as I presented, there are some experiments that studied the impacts of on chicken nutrition and with chicken challenged with some viruses, where really insect products mainly to decrease the mortalities by 20%. And of course, all the antimicrobial effects that we have seen in other species, especially, for instance, in Turkey, that is close to chicken, we can suspect they also exist in chicken. But yes, the work has been, several studies have been done on chickens also, and they also tend to reveal that we find antimicrobial effects in poultry too.
Yeah, if I can complement your answer, we have done this trial, this in vitro trial of antimicrobial effect on these bacteria, E. coli, Staphylococcus, and Salmonella. They are obviously a very pathogenic type of bacteria for not only for fish, but also for poultry. So in a very simple test in the lab using the diffusion disk, we can also notice very clearly the inhibition or the antimicrobial effect on this type of bacteria. If your question is regarding the use of insect meal in poultry, is growing a lot. There is several trials that is showing the benefit. It's of course is more challenging because we needed to replace soybean meal and the cost is more challenging, but the functional benefits for poultry, for broiler chicken and laying hens are also amazing, and at the same range of 25% replacement of the vegetable source, you can also have a functional benefits for these species too.
Another question. Are you able to extract the antimicrobial peptides, or do you plan so? So, okay, it depends if we answer at fundamental research scale or production scale. At fundamental research scale, some studies are ongoing to try to extract antimicrobial peptides and specifically target them against some bacteria or viruses. At production scale, it's not the goal now because it's still a very, very expensive method. But for production scale, I would say the goal is not to take some purified extract of even antimicrobial peptides. It would be rather, as Rafael presented it, to use the whole insect product as a whole. So it can include antimicrobial peptides, it can include it can include oil. We don't try to do separation. If we give insect meal, we give it as a whole. We don't try to extract specifically defensin, for instance.
Mm-hmm. But one question that also every time they ask is what is the mode of action of these insect products in different species, right? This is something that can be related with the, first of all, in our trial with shrimp, is related first of all with the nutrition. So we needed to understand quite well the gaps that we have in the formulation, the gaps or the limitations we have with insects to replace fish meal. Fish meal is of course a very premium product that contains lots of amino acids and protein, but is expensive and is not sustainable. It's really bad for the environment. So to replace successfully the fish meal in the diets, we needed to understand these gaps and formulate and use different additives such as amino acids or cholesterol, for example, to be more adaptable to shrimp. And then the mode of action is pretty much related with Charles just talked about the functional benefits of lauric acid, chitin, and these antimicrobial peptides that can give more resistance for the animals to deal with the challenges in the farm and the field. And what we have observed in our trial was that the survival rate was higher when we include higher doses of insect meal, and also we can observe a higher yields because the animals they do not die, so you have a higher yield per tank. So this is more or less the mode of action. these insect products can bring to the animals. All right. I don't know, Charles, do you have any more questions?
From the discussion, I don't have any more questions. Can you tell us if somebody wants to ask anything else? Maybe first? Ah, okay. Can I say?
Yes, I think this is about the product we tested, right? So yeah, sure, you can find the full amino acid. If you can write it to us, I can send to you the ingredient matrix where it comes with all the amino acid, the digestible amino acids, the energy content, all the nutrients that this insect meal can bring to the formula. And we are happy to share this with you. Yeah, for sure. It's important to mention that in the literature, you can find different information. Still, the black soldier fly, there is a lot of trials that is being published that the researchers, they produce their own black soldier flies, let's say in a backyard, using different substrate. And accordingly to use and the amino acid profile can change slightly if you change the substrate or the way you produce. So it's important to, this is a new ingredient we needed to understand, but there is a variability such as soybean meal. If you buy soybean meal from Europe or from China or from Brazil, they can have a slightly different profile. And it's the same for black soldier fly. So every company has their own process, or they use different substrates. So it's important to respect and to use the profile and analyze batch by batch what is the protein content and adapt to the formulation, do this precise formulation using the current profile from that specific product. But yeah, we are happy to send that information if you can write it an email for us. And then we have a question. What's your plan on the exuvia, the shedding cocoons? Do you understand this question, Charles?
Yes, I think it must deal with the fact that if we want to extract chitin, I guess, from exuviae, I think it must be this question. It must relate to this. Do you want to answer, Rafael?
Yeah. Well, the exuvia currently we are not using it, but I know that there is a lot of researcher that is using the exuvia to extract, for example, melanin. That is a natural black colorant that can be used also to produce computers parts, and also is a natural ingredient that can be used in the food. And also what I saw is there is a lot of people also looking for using this exuvia because of the chitin content to produce product for human nutrition, for animal nutrition, and also this chitin can be used to produce bioplastics. There is a high tendency to ban the use of plastic bags. We know that this is a big issue in our societies, the amount of plastic that we are generating, and the exuvia can be a substrate to produce bioplastics that is biodegradable and do not have an impact on the environment. Yeah, that's the information I have about the use of exuvia. And then the last question here is, to what extent are your two companies' products comparable beyond being from the same insect? Yeah, well, it's pretty much the same. Entofood and in Nutritech, we are both here in Malaysia. We are both using black soldier fly, and we are at the industrial scale, right? So it's important differentiate what is it, the industrial production of BSF and the backyard production, where you have a high variability because you have a high variability of the substrate. But yeah, I think we are very comparable. I don't know, Charles, if you have anything to.
No, I don't have any specific point to highlight. So maybe we can move to the next question. That was, do you have a side effect of lauric acid? We experienced the heat stress effect from C22. Honestly, regarding the side effect of lauric acid, I don't see any in particular in nutrition for black soldier fly. As I said, it's the most present in black soldier fly, the most present one. Like this, honestly, I don't see any side effect. Maybe Rafael, if you want.
Mm-hmm. No. I just saw trials that with a positive effect, because lauric acid can enter in the membrane and disrupt the bacteria, and there is a very well documented mode of action of lauric acid. I've never seen heat stress from coconut lauric acid. No, I didn't see that. If you can later share with us this, for me is a new.
Then any trials planned or done on functional effects on pet food, apart from the sustainability marketing aspect? Okay, not from at the production scale, but I know that in literature, pet food has been there is little evidence about pet food. Okay, it's a bit controversial because on the one hand, people are saying, okay, all the antimicrobial effects, we could find them, for instance, in dogs and cats. But on the other hand, it's not the same physiology at all, so we don't know. Some very recent works have, however, demonstrated, for instance, in dog, that insect products are very useful against arthritis problem. Like when dogs are getting older, they have a lot of arthritis problem regarding bones and articulations, and insect products have beneficial effects on this. So there is at least one very good point, I think. If you want to answer.
Yeah, in pet food, definitely. What we have seen also, the claim that pet food is looking for is the also hypoallergenic diets, where if they use like a vegetable kind of a source protein, this can have like a allergenic reaction in the animal. So insects, they do not cause this reaction. So it's also a very good ingredient to be used in pet food. That's what we have seen. They are exploring this angle. Yeah. Well, the other question we have here: do you have some info to share about insect meals nutritional data compared to fish meal? Yeah, sure. There is lots of information. We are glad to share this info with you. If we can also send the full matrix on this trial that we have done in shrimp vet in Vietnam. But obviously, the fish meal that we compare was very premium one with 67% of good protein. And usually our product contains 50% of good protein. So there is a big gap in terms of amino acids in crude protein that we have to formulate very carefully. We needed to be very, let's say, understand what is the gaps to make sure we have a precise nutrition, and then we can successfully change. It's not just replacing fish meal by itself, right? Taking 10 kilos from the formula and include 10 kilos of our insect meal. It needed to be reformulated. It needs to be combined with other sources of protein. In this case, in our case, we combine with soybean meal, which is less expensive than fish meal, of course. And with that, we were able to successfully implement this formula, and the result was a successful performance result. Comment from the audience, please be careful on the C12 dose. I do not know, but faced the problems many years ago. We met in broilers and a high mortality. Yeah, it's important that C12, the lauric acid, is a part of the insect oil, right? It's not that insect oil is pure lauric acid. There is, of course, in the market, different products that, for example, they can extract the lauric acid from palm oil, from coconut oil, and overall, the lauric acid is a component, and I don't know how these companies they isolate or they stabilize the lauric acid to be used in broilers. So, of course, there is always we needed to understand the how these products can be used. Yeah. Another question here: volumes are a key issue with alternative ingredients. Any info? Well, I suppose this question is about the volumes of the industry. I suppose, well, of course, this industry is just beginning. We have a huge gap in terms of protein. We were doing a calculation. If you want to replace 10% of the global fish meal demand, we needed to replicate our current factory 400 times, right? So just to take an idea on how big is the only one ingredient. So this industry is growing fast, but it's just we are just touching the surface. There is a need of a mass production, improvement of the getting gain on economy of scale, also be more effective. We needed to develop genetics, the understanding of nutrition. So all of these will help us to produce more from less and be more affordable during time. This is a technology that is just beginning, in my opinion.
Just to complete, Rafael, as I said, for instance, for the pet food market, only for the protein market, sorry, they are projecting a multiplication by 50 of the volumes in 10 years. So, of course, the industry is still young, but can I say, we can be very optimistic.
Yeah, sure. We as we are growing and evolving, we have a better understanding of the substrates and production in different areas, right? So I see black soldier fly growing in every countries, being able to use and upcycle food waste that was supposed to be disposing in the landfills. So there is a lot of possibilities to growing in terms of volume and producing locally. Yeah. The other point that is super important is that we do not depending on the commodities fluctuation, so the price is stable during the whole year. And this is also we noticed during the pandemic situation, the prices was getting higher and higher, high fluctuations, and this is not good for stabilize the industry. And the BSF, the insects can stabilize by producing locally and do not fluctuate in the price. Do you guys have any more questions for us? No, I think we are right on time. No more questions. Okay, good.
Yes, of course, you can contact us for the VConnect profile.
Yes, we are open to hear if you have more questions. And yeah, thank you very much for your attention. Thanks, Charles.
Yes, thank you very much to everybody. Thank you, Rafael.
Thank you. Bye-bye. Have a good one.
Thank you. Goodbye.